In the last video, we looked at how to perform what we called a one-trait cross. So we were creating a Punnett square, looking at only a single trait at a time. But I think you can imagine that our entire genome is made up of more than one trait, so this gets more and more complicated the more traits that you consider to predict what the entire genotype of a whole genome would be. Don't worry, I'm not going to be asking you to predict the genotype of an entire genome, but I do want to do a two-trait cross to demonstrate that that process is going to be the same to create the Punnett square no matter how many traits you're looking at. So we're going to do a two-trait cross and predict the possible phenotypes and genotypes of the offspring. The first trait here we're already familiar with. We know that having freckles or not is a dominant trait determined by the big F allele being dominant, leading to the phenotype of having freckles. And the little f allele is recessive, leading to the phenotype of having no freckles. In this case, both mom and dad are heterozygous, big F, little f. Trait 2 is going to be having a widow's peak or not. Again, a dominant trait. So big W allele is dominant, leading to the condition or phenotype of having a widow's peak. And little w allele is recessive, leading to the phenotype of having no widow's peak. If you don't know, a widow's peak is this peaked hairline. There we go. This peaked hairline right here. So if you were homozygous recessive, little w, you would have a straight hairline. If you were heterozygous or homozygous dominant, you would have a widow's or a peaked hairline. Again, in this case, both mom and dad are both heterozygous, big W, little w. To perform our Punnett square, the process is the same as when we were doing a one-trait cross. You're going to first determine the possible gamete combinations of mom and put them on one side, what would be in the eggs, and the possible gamete combination of dad, what would be in the sperm, and put them along the other side. And then you would match them up. Big F from dad, big F from mom, big W from dad, big W from mom. And in this case, though, there are more than 4 squares, like we saw in the one-trait cross. So for a two-trait cross, you would have a total of 16 squares. And so if you were asked for a probability of being both freckled and having a widow's peak, you would count up all of the squares that had at least one big F and at least one big W. And if you added them all, in this case, they're in the darker blue here, and that would be 9 out of 16. So the chance of having an offspring that is both freckled and having a widow's peak is 9 out of 16. The biggest thing that students have issues with when creating this two-trait cross is coming up with how to determine the gametes to put on both sides for mom and dad. So we can use what we call the FOIL method to do that. First, outside, inside, last. First, outside, inside, last. So in order to do that, you're going to write out the genotype of one of the parents. We'll do mom in this case, although for this example, they're actually both heterozygous for both traits, so their gametes come out being the same. So you're going to write out her entire genotype, and then you're going to take the first 2 from each trait. Big F, big W first. And put that in that first square. Outside, big F, little w, and put that on the second square. Inside, little f, big W, put that on the third square. Last, little f, little w, and make that the fourth square. And then, of course, dad's is the same, so you would put those also along the top. So that is how FOIL method is how you're determining what their gametes will be. That one's already filled in for you. Let's go through another example, a new example of a two-trait cross, and how to fill in the Punnett square for if we need a little bit more practice with that. And your lab, of course, will give you more practice with that for this week as well. Trait 1 for this cross is going to be hair color, which is a dominant allele. This is kind of a hypothetical scenario because hair color is something that's determined by multiple alleles. But for our purposes here, we're going to give it the B allele. And we're going to call the B allele dominant, leading to brown hair, and the little b allele recessive, leading to blonde hair. And then our trait 2 is having either unattached or attached earlobes. That's whether or not your earlobe dangles down a little bit or is attached and goes straight to the side of your face. And this one actually is a completely dominant trait. So the big E allele is dominant, leading to unattached earlobes, having a little bit of a dangle. And little e allele is recessive, leading to attached earlobes, where it goes directly to the edge of your face. For our example here, both mom and dad, again, are heterozygous for both of these traits. So the possible gametes for mom will be the same as the possible gametes for dad. So if you were going to perform the FOIL method, the first thing that you would do is write out mom's entire genotype, big B, little b, big E, little e. F stands for first, so big B, big E. O stands for outside, big B, little e. I stands for inside, little b, big E. And last, L stands for last, little b, little e. And then in this example, dad is also heterozygous for both traits. So you'd only have to perform the FOIL method one time to determine those gametes. So you would take these 4 gametes to create your Punnett square, and you're going to put mom's along this side, and dad's along the top, again being the exact same. That was terrible. And then I'm not going to make you sit here and watch me fill in the entire Punnett square on this video. So let's pick a random square here just so we can demonstrate how to fill that in. So if we're going to fill in this square, you would take big B from mom, big B from dad, little e from mom, and little e from dad. So this potential offspring's genotype is homozygous dominant for hair color and homozygous recessive for earlobe shape, attached or unattached earlobes. So based on that genotype, big B, little e, what would be that individual's phenotype? What they look like? Hopefully you said brown hair, because big B is dominant, with attached earlobes, because that person was homozygous recessive, and the attached earlobes is the recessive trait. But that would be one example of how you could use that Punnett square to answer a question about those potential genotypes and phenotypes of their offspring. So that is 2 trait crosses. Now, all of the examples that we have seen so far have been fairly simple in that the traits show complete dominance, which means that one allele is completely dominant to the other and will always cancel the other out. However, there are some traits that are expressed by codominant alleles, which means that both of the alleles will express a specific protein at the same time, resulting in more complicated expression. So they won't cancel each other out, but rather they will both be expressed. Having an AB blood type is an example of this, because the A and B alleles for blood type are considered codominant. So A, B, and O are our 3 alleles that are possible for determining blood type, and they indicate the polysaccharides that are going to be lining on the surface of our red blood cells. And if you have an AB blood type, so you have an A and a B allele, then you're going to produce both of those kinds of polysaccharides. One is not going to cancel out the other. They will both be expressed. That's an example of codominance. Within blood type, though, you also have an example of dominance, complete dominance, because A and B alleles are both completely dominant to the O allele. So you have to know which ones are codominant and which ones are completely dominant. And that one is a bit tricky. You get a chance to explore that a little bit more in your lab for this week. So that's an example of codominance. Alleles can also display incomplete dominance. And in incomplete dominance, the expression of the trait in a heterozygous individual, so having one copy of both alleles, will be somewhere in between the expression of a homozygous dominant or a homozygous recessive. So whereas in codominance, both traits were expressed equally at the same time, in incomplete dominance, neither trait is expressed, but rather it's kind of a mixed phenotype. So flower color is a really common example of that. Having an R allele for color, being homozygous for R will lead to a red flower. Being homozygous for white will lead to a white flower. But instead of being like striped red and white, like you would expect from maybe a codominant situation, this phenotype of the heterozygote is pink, a mixed phenotype. Neither one is expressed over the other. Other kind of oddball examples would include genes that are considered sex-linked. So these are going to be genes that are housed primarily on the X chromosome, and they're considered sex-linked because in males, traits that are determined by genes on the X chromosome can only be determined by that single allele because they're only going to have one copy of it. The Y chromosome is much, much smaller and doesn't contain the same genetic information as the X chromosome, and really only contains the information needed to produce those extra testosterones or male sex hormones. So anything that's coded for on the X allele is going to be determined by only the one allele that they have on their X chromosome. An example of that is going to be color blindness. That's one of the alleles that is housed on the X chromosome. And our last example is polygenic inheritance. And these are going to be traits that are determined by multiple genes working together. So examples of that would be skin color and height. Those are kind of the most common examples of polygenic traits. But I kind of just wanted to show you this because this is still a Punnett square that is being performed, obviously probably by a computer and not by somebody drawing it out at home like you're going to be doing. But it's the same process that you can go through. As long as you know all of the traits, all of the genes that lead to that trait or contribute to that trait, then you can perform a Punnett square to figure out what the possibilities are. And polygenic traits are just determined by multiple genes. Sorry, lost my train of thought. Okay, that is it for our discussion of genetics. If you have any questions at all, don't hesitate to reach out. I am always here.